Thank you, Manuel and Nayantara, for the invitation. Can you see my screen? I guess you can. So, today I want to talk to you um, about comparing rhythmic patterns, which is the next level of analysis from the kinds of things Tanya spoke about in the previous talk. And I'm at the Institute for Theoretical Biology at Humboldt University in Berlin. And just to give you a brief introduction, in my group, we use a combination of mathematical modeling and data analysis to answer questions in circadian biology and circadian medicine. And so today, one of the topic today is controlled experiments in circadian biology. So one of the common paradigms for understanding the functioning of a system is to observe the output of the system under a control condition and after a, a designed perturbation, treatment, or intervention. And by comparing the responses under the two conditions, by observing what are the differences, one can gain insight into how the system functions. And in our context today, these treatments or perturbations might be variations in the Zeitgeber input to the system, or they could be molecular in nature, sort of knockouts, knockdowns, overexpression, and so on. In terms of outputs, we already heard several today. It could be at the whole organism level, at the systemic level. It could be activity, behavior at the tissue level, or it could be individual components of the molecular clock. And if one uses a high throughput assay, one might actually then screen multiple of these molecular clock components in one go. And that's going to be the focus of my talk today. So once one has assayed the entire a large number of molecular species under the control and experimental condition, the intuitive approach would be to ask the question, how has this intervention affected the circadian system? So then one chooses your favorite rhythm analysis tool from, for example, this many that Tanya introduced you in the previous talk. And then we do a rhythmicity analysis. And for this hypothetical case, we conclude that this gene expression or this gene lost rhythmicity after the intervention, because the p-value for rhythmicity was significant in the control condition, but not in the experimental or the intervention condition. And now when we go to an entire high throughput study, one has then a list of features. So features that were rhythmic under the control condition and those that were rhythmic under the treatment condition. And then one can use a Venn diagram or a set theoretic approach to ask what is rhythmic in both conditions, what is rhythmic only in the control or in the treatment condition. And just to clarify, by features here, I mean any species that I assay using my high throughput approach is considered a feature. So it could be genes, it could be transcripts, proteins, and so on. And of course, when we do the rhythmicity analysis across the entire set of features, one has to take care of multiple testing and adjust the p-values accordingly. And I'm assuming that has been done. And under this controlled experimental paradigm, what we are often interested in is in features that change rhythmicity between the control and the treatment condition. That is, we are interested in features that fall in these hatched regions in the Venn diagram. So let's evaluate how this intuitive approach works. Okay. So I'm going to do this using two artificial experiments that I'm going to construct. In the first, that I consider a negative control, I took the liver transcriptome data, which was measured every hour over 2 days. So that's 48 samples. And I split these 48 samples into two groups of even and odd samples and call them two new data sets, even and odd. And the time series for one of the genes is shown on the right here with the even and odd samples marked out. And then I do an artificial experiment where I compare what is different between the even and odd samples. Of course, we know from the biology that both these samples, both these data sets are nothing but downsampled versions of the original data. That is the original data at lower time resolution. So we don't expect to see anything, or biologically, we don't expect any difference between the odd and the even samples. But if I follow this Venn diagram approach I outlined in the previous slide, we observe that more than 50% of the rhythmic transcripts are actually classified as being rhythmic only in odd or only in even. One might then think, oh, maybe this is because one was very relaxed with the rhythmicity analysis, and we could use a much more stringent false discovery rate cutoff. And we observe that the problem still remains. It's just that we make fewer, we call fewer transcripts to be rhythmic only in odd or only even, but at the cost of having fewer rhythmic transcripts on the whole. And in fact, if we sweep across different thresholds for calling rhythmicity, we observe between 40 and 50% of rhythmic transcripts are claimed by this approach to be rhythmic only in odd or only even. This is clearly wrong. In order to complete this analysis, what we did was we took the odd data set and shifted the rhythms of 500 selected transcripts. That is, we changed their amplitude and phase so that we had a known set of true positives in the data. So genes that we know to have altered rhythmicity. And then we redid the Venn diagram approach. So the number of true positives that we recovered correctly are given in the square brackets. And you can see that between them, we only recovered about 200 of the 500, but we still made many false positive differential rhythmicity calls. That is, we called many transcripts to be rhythmic only in the even or only in the odd data set. And one can then quantify this using something called a precision recall curve, which maps how many of the true differentially rhythmic transcripts that we do recover by the algorithm versus in any given call, how many of our calls are actually correct. And we observe that we never get more than 20% of our calls right for any choice of threshold, of FDR threshold. In other words, 80% of our differential rhythmicity calls are wrong, right? Which is terrible. So to summarize, this Venn diagram based approach makes many false positive or identifies many false positive differentially rhythmic features, both in the presence of true differentially rhythmic features and in the absence of those. So why are then Venn diagrams misleading? So let's take an example with an example. So the black dots represent the true classification of 4 different transcripts, for example. So here we assume that we have a crystal ball and we know exactly which group each of these 4 transcripts belong to. Okay, and now, of course, no rhythmicity analysis tool is perfect, so it's going to make mistakes. So, for example, with this particular transcript, which is actually rhythmic in both, if we make a false negative in the control group, then this is then classified by the algorithm to be only rhythmic in the treatment group, even though it's rhythmic in both. Similarly, if we take something which is rhythmic only in the control with a false positive, this can go into the rhythmic in both category. And further, you can have false negatives that take something which is rhythmic in one category completely out of this analysis and can also bring in, due to a false positive, into the analysis. However, in most high throughput studies, that is studies where you have many features that one is testing, one always tries to control the number of false positives at the cost of accepting false negatives, right? That we don't want to make a false discovery. We don't mind if we miss some discoveries, but we don't want to make any false discovery. And so, as you can see here, since you make a lot more false negatives than false positives, you generally tend to take features that are classified as rhythmic in both out of this into one of the differentially rhythmic groups rather than the reverse. And this is the reason that this Venn diagram based approach overestimates the amount of circadian remodeling or reprogramming, as it's often termed in the literature. And this has been used extensively in many high impact studies in high impact journals. So what is then the solution? And as Gregory said, this is part self-promotion. And there are two ways to get around this problem. But the general idea is one should reformulate the question, and instead of asking what is rhythmic in A and what is rhythmic in B, or what is rhythmic in the control and what is rhythmic in the treatment group, one has to ask the question, are the circadian parameters different between the two groups? Right? So one has so there is a hypothesis testing way of formulating this, where we say the null hypothesis is that the amplitude and phase of this particular feature is the same in the two groups. And then one can come up with a test and a p-value for this differential rhythmicity, and that way control the number of false positives. The other approach completely sets aside this p-value idea, but instead fits many different groups of patterns. So you could have, which are shown here, 4 of those, plus the category that that the feature is not rhythmic in both conditions. So you could have a feature that's rhythmic in not rhythmic in the control, rhythmic in the treatment, rhythmic in the control, not rhythmic in the treatment, and so on. And one tries to classify every feature into one of these groups and uses an information theoretic criterion to decide if this classification we are confident enough in the classification. And this is available as an R package in my GitHub page. And I completely second Gregory's idea about, what Gregory said about open source software and making everything available. In fact, code for all my publications are also available on my GitHub page. So in order to use this package, all we need is a data matrix where we have the different samples in the columns and the different features in the rows, and an experimental design matrix which describes what the different samples contain. So it describes which group the sample belongs to, the time point that sample belongs to, and if it comes from a replicate. And since this is a very flexible way of defining your data, this can take care of missing samples, unbalanced design, unequally spaced samples, and so on. And in order to run this analysis, you just call a single R function with these two pieces of information, and one can choose the method one wants, and one can either just retrieve the classification of the different features into the different groups, or can also get p-values and estimates of the amplitudes and the phases under the different conditions. So in this work, we also went back and reanalyzed these high impact studies that use this erroneous Venn diagram analysis. And you can see here the first column in bold is the reported fraction of circadian transcripts. So these are all transcriptomic data sets. So showed that more than 70% of the rhythmic transcriptome was altered by their treatment, which involved, for example, changes involved high-fat diet or a ketogenic diet or modification of certain cellular pathways, for example. But if you do this analysis correctly using compareRhythms, we see that except for this ketogenic diet, we get less than 5% of the transcriptome actually being altered by these interventions. Of course, the consequences of this overestimation we discuss in detail in the paper, and I don't want to kind of get into this in the talk here. So the take-home messages from today are, which is actually broader than just rhythmicity analysis, is do not, if you want to make an inference, run a statistical test directly for that inference that you want to make, and make a conclusion based on that. Do not do multiple tests and draw multiple separate inferences and then combine these to make an inference. And that generally is always going to give you a lot more false positives. And be very wary about using Venn diagrams except as a visualization tool. And this, whatever I spoke about and the description of the package is available in this publication list here. And for those who are interested in this problem, as I mentioned, in fact, everything that I spoke to you about works for anything else you compare between two groups. And this really great paper in Nature Neuroscience talks about this in the broader context. And they, in fact, surveyed many nature science papers. And they found that 50% of the papers they surveyed actually did this analysis incorrectly. So I thank you for your attention, and I'm happy to take some questions.
Wow. Okay, Bharath, that was really a very interesting talk. And it raises a lot of questions about statistical analysis that I'm sure all of us wrestle with. But first, let me open the floor to the audience. Do you have any questions? And please type in the Q&A. And I will save the really intense discussion questions for the panel section. I have more of a technical question, Bharath. So the analysis package and your code that you shared, all of the data you showed were mostly transcriptomic data. So is this very specific to transcriptomic data, or could it be used for the broader analysis of circadian rhythms in physiological data? Things like cognitive data or just actigraphy data that a lot of the talks focused on today?
So, so the thing is, the way, so in my package really uses just kind of cosigner analysis, right? So just fitting sine curves to the data. So in that sense, it's very generic. You can use it with any tool. However, and actually, if you go through the help file, I actually described, so for transcriptomic data, there are some special settings that let you do better than if you just did it in a kind of data agnostic way. But of course, there are certain settings in which you can run it on pretty much any data. It's not dependent necessarily on it being an omics data set, although the paper itself takes examples only from the omics context.
Okay,